Imagine a technology that sits right at the crossroads of genetics, IVF, and cutting-edge molecular biology. That technology is preimplantation genetic diagnosis, or PGD for short. It first came on the scene back in the late 1980s, and it completely changed the game, offering a powerful new alternative to traditional prenatal testing by letting us screen for inherited diseases before pregnancy even starts.
So, here's our game plan for this deep dive. First, we'll tackle the basic genetics you need to know. Then, we'll walk through the actual clinical process and see how the technology behind it has evolved. After that, we're going to jump right into the big controversies swirling around the field, and we'll wrap up by looking at the future and the tough ethical questions we're all facing.
Okay, so to really get a grip on why PGD is such a big deal, we've got to rewind a bit and talk about some basic genetics. Let's quickly go over the three main ways that single-gene diseases get passed down through families.
First up, we have autosomal recessive inheritance. Now, in this scenario, both parents are what we call carriers. They each have one copy of a mutated gene, but they don't show any signs of the disease themselves. The catch is, their children have a 1 in 4 chance of inheriting both of those mutated copies and actually having the disease. Cystic fibrosis is probably the most well-known example of this.
Next, you've got autosomal dominant inheritance, and this one has a totally different risk profile. Here, you only need one copy of the mutated gene for the disease to show up. So if one parent has the condition, there's a 50-50 chance they'll pass it on to their child in any given pregnancy. We see this pattern a lot in diseases that show up later in life, like Huntington's disease.
And finally, there's X-linked inheritance. As the name suggests, the gene mutation is on the X chromosome. Now, because males only have one X, which they get from their mom, a carrier mother has a 50% chance of passing that affected X chromosome to her son, and if he gets it, he'll have the disease. This is the genetic story behind conditions like Duchenne muscular dystrophy and hemophilia.
So when you understand these inheritance patterns, you can really start to appreciate the incredibly tough spot that couples with these genetic risks used to find themselves in. Discovering a fetus was affected mid-pregnancy meant facing a truly heartbreaking decision. PGD was created to offer a completely different way forward.
And this, right here, is the fundamental shift, the real game changer that PGD brought to the table. Traditional testing happens when you're already pregnant. PGD moves that whole diagnostic process way earlier, into the IVF lab, before a pregnancy even begins. It allows couples to select an embryo that's free of the specific disease from the very start. This was a revolutionary idea designed to get around a huge ethical dilemma for so many families. So how does this actually work in practice? Well, it's not a simple blood test, that's for sure. It's a really complex, multi-step procedure that requires this incredible teamwork between fertility doctors, embryologists, and geneticists, all working in perfect sync.
The whole thing boils down to 4 main stages. It all kicks off with a standard IVF cycle to create the embryos. Then, at a very specific time in their development, a tiny biopsy is done to get a sample of genetic material. Those cells get sent off for analysis. And based on those results, the embryos that are diagnosed as unaffected are chosen to be transferred back into the uterus.
Now, that biopsy step is super critical, and there are a few ways to do it. You have polar body biopsy, which is done on the egg itself, but the big catch is it only tells you about the mother's genetic contribution. Then there's cleavage stage biopsy on day 3. That used to be the standard, but it has a high risk of misdiagnosis because of something called mosaicism. Today, the gold standard is blastocyst biopsy on day 5 or 6. You get more cells to test, but here's the tricky part. These cells are from the outer layer, which becomes the placenta, and they might not be a perfect match for the inner cells that actually become the baby.
So once you have those biopsy cells, you have to analyze them. And the technology for doing this has just been moving at lightning speed. It's like going from trying to solve a puzzle with only a handful of pieces to suddenly having the whole picture on the box top to look at.
Back in the 90s, the field was using tools like FISH and PCR. They were groundbreaking for their time, but they were also limited and could make mistakes. Then, in the 2000s, Array-CGH came along and let us screen all 24 human chromosomes at once, a huge leap forward. And today, we have next-generation sequencing, or NGS. This is an absolute powerhouse that lets us look for chromosomal errors, single-gene diseases, and other structural problems, all from that one tiny biopsy and with incredible accuracy.
But even with these amazing tools, there are still technical gremlins to watch out for. One of the biggest, especially for those older PCR-based tests, is something called allele dropout. Basically what happens is the test fails to see one of the 2 copies of a gene.
And if the copy it misses is the one with mutation, you could have a catastrophic misdiagnosis. An affected embryo gets mistakenly called healthy. Modern testing has clever ways to try and prevent this, but it's a risk that's always there.
And all this amazing technology brings us to, well, probably the biggest and most heated debate in the entire field. The controversy isn't really about using PGD for these severe single-gene diseases we've talked about. No, it's about using it to screen all embryos for chromosomal errors in the general IVF population. This practice is now called PGTA, but for years everyone knew it as PGS.
This is the million-dollar question. On the surface, the logic seems rock solid, right? We know that having the wrong number of chromosomes is a huge reason why embryos don't implant or lead to miscarriage. So if we only transfer the ones that are chromosomally normal, or euploid, we should get better results. The problem is, the actual clinical data has been way, way messier than that.
So why is this so controversial? Well, there are a bunch of strong arguments against using it routinely. For starters, it doesn't fix or improve an embryo. It's just a selection tool. The biopsy itself is invasive. Having normal chromosomes doesn't guarantee a baby. And like we said, that biopsy of the outer layer might not tell you the real story. On top of that, it makes an already expensive IVF cycle even more costly. Some data even suggests a link to a higher risk of preeclampsia in the mother. But the biggest biological wrench in the works is a phenomenon called mosaicism.
Think of a mosaic embryo like a patchwork quilt. It's made up of a mix of different cell populations, some with the normal number of chromosomes and some with an abnormal number. We now know that this is actually super common in early human embryos, and you can see how this makes diagnosis a nightmare. If you take a biopsy of just a few cells, you might be grabbing a piece from an abnormal patch of what is otherwise a mostly normal embryo.
But here is where the story gets absolutely fascinating. A growing body of research from animal models to actual clinical results has shown that many of these mosaic embryos can do something incredible. They can self-correct. The cells that are destined to become the fetus can actually push out or get rid of the abnormal cells, leading to the birth of a perfectly healthy, chromosomally normal baby. This discovery completely shakes the foundations of PGS and the old practice of just discarding any embryo labeled abnormal.
This deeper understanding of how embryos actually work leads us right to the future of this technology, and the really complex ethical territory we're now having to figure out how to navigate.
The designer baby fear. That has always been the big, scary headline, right? And look, it's a valid concern we need to talk about as a society. But the reality is that in many places, like the UK, there are strict regulatory bodies that have flat out banned using this technology for non-medical reasons. Plus, given how invasive, expensive, and uncertain the whole process is, it's a really impractical way to go about selecting for trades. And we also have to be really realistic about the practical hurdles. PGD is not a magic bullet. Misdiagnosis, while rare, can still happen because of those biological and technical issues we talked about. The success rate, the chance of taking home a baby, isn't necessarily any higher than with standard IVF. And the cost, it can be staggering, sometimes doubling the price of a treatment cycle, which puts it completely out of reach for a lot of people.
So we end up here with this really profound question. As our ability to read an embryo's entire genetic blueprint gets more and more powerful, we're forced to ask, what does it actually mean for an embryo to be healthy? Is it about not having one specific disease gene? Is it about having a perfect set of chromosomes? Or is it something more? Something about having the potential to fix itself and grow into a healthy child? The technology is racing ahead, but these are the fundamental questions that are going to stick with us for a long, long time.